Hello, everyone. My name is Alejandro Torres. I'm a third-year PhD student, and today I'm going to share with you some of my research. So it's titled BDNF levels affected by the synthetic cannabinoid WIN55 212-2 in adolescent rats. Cannabinoids are molecules that bind to endocannabinoid receptors CB1 and CB2 present in the central and peripheral nervous system. The synthetic CB1 and CB2 receptor agonist WIN55 emulates the effects of delta-9 tetrahydrocannabinol, THC, the psychoactive component of cannabis plant. Endocannabinoids, naturally found anandamide and 2-AG, and the neurotrophin brain-derived neurotrophic factor, BDNF, are potent neuromodulators that play critical roles during the development, physiological processes, endocrine homeostasis, stress anxiety response, and neuroplasticity. BDNF is found in two different forms. The precursor pro-BDNF binds to P75, causing a synaptic pruning or apoptosis. However, mature BDNF binds to TEOR-kappa B receptor, signaling cell survival and neurogenesis. Disruption of either BDNF or endocannabinoid signaling is associated with an overlapping set of neurologic and psychiatric diseases. The chronic use of exogenous synthetic cannabinoids during adolescence, a vulnerable stage for brain development, might affect or alter the homeostasis and neuroplasticity-dependent BDNF. The adolescent brain is an important topic for us because a great amount of neuroplasticity happens during this stage, important for cognitive and psychosocial interaction and goal-driven behaviors into adulthood. The hippocampus is a brain structure that is deeply involved in memory formation and retrieval. Animal studies suggest that acute cannabis administration disrupts the hippocampal-dependent spatial learning in rats. Periconductal gray, or PAG, is an area that contributes to the modulation of anxiety, fear, and nociception, all which may produce physical discomfort, and these can be seen on withdrawals linked with the chronic exposure of opioid abuse. How we did our experiments was by employing an animal model consisting of spraguidally male rats, and we gave them 2 times daily intraperitoneal injections of WIN55 or the saline vehicle solution for 5 different sessions every 24 hours on space in each of the injections. We then extracted on the last day the brain and the blood, and we dissected the brains on different areas that we were interested on, as the prefrontal cortex, the hippocampus of the PAG. We also homogenized these tissues and collected the serum and run them in a ELISA immunoassay kit to detect the BDNF concentrations. What we observed is that there were significant increases in the PAG for pro and mature BDNF levels after the treatment. And on the serum, we can only observe pro-BDNF increases after the WIN55 treatment, but we did not see any increases on the mature BDNF. But for the hippocampus, we actually saw the mature levels were increased. However, for the prefrontal cortex, there was no significant increase on the mature BDNF. The chronic exposure of synthetic cannabinoid during adolescence, now we know that modifies pro and mature BDNF ratio in the dorsal, PAG, and periphery, and also increases mature BDNF levels in the hippocampus, suggesting that pro-BDNF and mature BDNF ratio are involved in endocannabinoid-mediated adolescent plasticity. We would like to measure the levels of the pro-BDNF from prefrontal cortex, hippocampus, and the cerebellum of those treated animals. Also, we want to investigate what are the effects of the administration of THC on BDNF expression using a mutant model of BDNF. And also, we want to study how the microbiome gut-brain axis is affected due to ingestion of administration of cannabis-derived products. These are my references, and I want to thank you for your time. And if you have any questions, feel free to email me. Thank you.